This article is about the genus. For the disease, see shigellosis. Shigella is a genus of gram-negative, facultative anaerobic, nonspore-forming, nonmotile, rod-shaped bacteria closely related to Salmonella. The genus is named after Kiyoshi Shiga, who first discovered it in 1897. The causative agent of human shigellosis, Shigella causes disease in primates, but not in other mammals. It is only naturally found in humans and apes. During infection, it typically causes dysentery. Shigella is one of the leading bacterial causes of diarrhea worldwide. Insufficient data exist, but conservative estimates suggest Shigella causes about 90 million cases of severe dysentery, with at least 100,000 of these resulting in death, each year, mostly among children in the developing world. Classification: Shigella species are classified by 4 SRO groups: SRO Group A, S. dysenterii, SRO Group B, S. flexneri, SRO Group C, S. boydii, SRO Group D, S. sonu. Groups or Euro-C are physiologically similar. S. sonu can be differentiated on the basis of biochemical metabolism assays. 3 Shigella groups are the major disease-causing species. S. flexneri is the most frequently isolated species worldwide, and accounts for 60% of cases in the developing world. S. sonu causes 77% of cases in the developed world, compared to only 15% of cases in the developing world. And S. dysenterii is usually the cause of epidemics of dysentery, particularly in confined populations such as refugee camps. Each of the Shigella genomes includes a virulence plasmid that encodes conserved primary virulence determinants. The Shigella chromosomes share most of their genes with those of E. coli K12 strain MG1655. Phylogenetic studies indicate Shigella is more appropriately treated as subgenus of Escherichia, and that certain strains generally considered E. coli a such as E. coli 0157, H7 a are better placed in Shigella. Pathogenesis: Shigella infection is typically by ingestion. Depending on age and condition of the host, fewer than 100 bacterial cells can be enough to cause an infection. Shigella causes dysentery that results in the destruction of the epithelial cells of the intestinal mucosa in the cecum and rectum. Some strains produce the intratoxin Shiga toxin, which is similar to the verotoxin of E. coli 0157, H7 and other verotoxin-producing E. coli. Both Shiga toxin and verotoxin are associated with causing hemolytic uremic syndrome. As noted above, these supposed E. coli strains are at least in part actually more closely related to Shigella than to the typical E. coli. Shigella species invade the host through the M cells interspersed in the gut epithelia of the small intestine, as they do not interact with the apical surface of epithelial cells, preferring the basolateral side. Shigella uses a type II secretion system, which acts as a biological syringe to translocate toxic effector proteins to the target human cell. The effector proteins can alter the metabolism of the target cell, for instance leading to the lysis of vacuolar membranes or reorganization of actin polymerization to facilitate intracellular motility of Shigella bacteria inside the host cell. For instance, the ICSA effector protein triggers actin reorganization by NWASP recruitment of ARP 2/3 complexes, helping cell-to-cell spread. After invasion, Shigella cells multiply intracellularly and spread to neighboring epithelial cells, resulting in tissue destruction and characteristic pathology of shigellosis. The most common symptoms are diarrhea, fever, nausea, vomiting, stomach cramps, and flatulence. It is also commonly known to cause large and painful bowel movements. The stool may contain blood, mucus, or pus. Hence, Shigella cells may cause dysentery. In rare cases, young children may have seizures. Symptoms can take as long as a week to appear, but most often begin 2 to 4 days after ingestion. Symptoms usually last for several days, but can last for weeks. Shigella is implicated as one of the pathogenic causes of reactive arthritis worldwide. Diagnosis: the diagnosis of shigellosis is made by isolating the organism from diarrheal fecal sample cultures. Shigella species are negative for motility and are generally not lactose fermenters, but S. sonu can ferment lactose. They typically do not produce gas from carbohydrates and tend to be overall biochemically inert. Shigella should also be urea hydrolysis negative. When inoculated to a triple sugar ion slant, they react as follows: Ka, gas, and H2S. Indole reactions are mixed, positive and negative, with the exception of S. sonu, which is always indole negative. Growth on hectin enteric agar will produce bluish-green colonies for Shigella and bluish-green colonies with black centers for Salmonella. Prevention and treatment: handwashing before handling food and thoroughly cooking all food before eating decreases the risk of getting shigellosis. Severe dysentery can be treated with ampicillin, TMP-SMX, or fluoroquinolones, such as ciprofloxacin, and of course rehydration. Medical treatment should only be used in severe cases or for certain populations with mild symptoms. Antibiotics are usually avoided in mild cases because some Shigella species are resistant to antibiotics, and their use may make the bacteria even more resistant. Antidiarrheal agents may worsen the sickness, and should be avoided. For Shigella-associated diarrhea, antibiotics shorten the length of infection. Currently, no licensed vaccine targeting Shigella exists. Shigella has been a long-standing World Health Organization target for vaccine development, and sharp declines in age-specific diarrhea dysentery attack rates for this pathogen indicate natural immunity does develop following exposure. Thus, vaccination to prevent the disease should be feasible. Several vaccine candidates for Shigella are in various stages of development. See also, diarrheal diseases, enterotoxigenic E. coli, infectious diarrhea, traveler's diarrhea, references. External links: Shigella genomes and related information at Patrick, a bioinformatics resource center funded by NIAID, World Health Organization, Shigella datasheet. New Zealand Food Safety Authority. Retrieved February 22, 2012. A Vaccine Resource Library, Shigellosis and enterotoxigenic Escherichia coli.